So far we have been discussing about tricarboxylic acid cycle. So where pyruvate it gets converted to carbon dioxide and water, and also we have seen how oxaloacetate has been regenerated. So now we move on to the next part where we will also discuss about the step-by-step events taking place in this particular pathway, and also the main step that regulate or control the entire cycle. So the first step is where the methyl carbon of acetyl-CoA it's linked with the oxaloacetate, and it again results in the formation of a 6-carbon compound called citrate, and this is basically a condensation reaction. And the CoA enzyme, which contains the sulfhydryl group, and it is being regenerated in the step 1. Okay, so whatever CoA which is being transferred to the pyruvate, it is again being regenerated here during this process, and this CoA which is liberated during this process, it is again used up by the pyruvate molecule. So this particular reaction is catalyzed by the enzyme citrate synthase, and then the second reaction it's catalyzed by the enzyme aconitase. Here the citrate it loses a water molecule, that is it is dehydrated for the first step, and then again it is a water molecule is added to this particular intermediate compound, and it results in the formation of isocitrate. Isocitrate is a isomer of citrate, and the intermediate compound that is formed here is this aconitate, which is an unstable compound. And then the third reaction, it is being catalyzed by isocitrate dehydrogenase. Here the isocitrate it gets converted to alpha ketoglutarate, which is a 5-carbon compound, by the process of oxidative decarboxylation. So here we can see that one molecule of carbon dioxide has been evolved, and also a hydrogen molecule is also removed from the substrate. So this hydrogen, whatever is being formed, it liberated, it is getting converted or transferred to the electron acceptor. Here NAD+, it serves as the electron acceptor, and it gets converted to NADH. That is, it is getting reduced. NAD+ gets reduced to NADH. And then the fourth reaction is where the alpha ketoglutarate it gets converted to succinyl-CoA, and this is again catalyzed by the enzyme alpha ketoglutarate dehydrogenase. And here again one more molecule of carbon dioxide is removed, and since this is a dehydrogenase enzyme, it again loses a electron, and that is being accepted by this NAD+, which serves as the electron acceptor. And this enzyme alpha ketoglutarate it again resembles this enzyme pyruvate dehydrogenase in its structure and also in its function. It is also multi-enzyme complex, just like the pyruvate dehydrogenase. And for this step, succinyl-CoA it acts as an inhibitor. And then the next reaction is where the succinyl-CoA it gets converted to form succinate by the enzyme succinyl-CoA synthetase. So I already told you that the difference between synthase and synthetase is, in synthetase enzyme the entire reaction will be carried out by the ATP or any energy molecule will be there. But for reactions which are catalyzed by synthesis, energy molecules are not required. So here the driving force is going to be the GTP, and this there is also one more step in which this GTP can be converted to ATP by the enzyme nucleoside diphosphate kinase. Okay, and then the next reaction is where the succinate it gets oxidized to fumarate by the enzyme succinate dehydrogenase, and this is followed by the fumarate which gets converted to malate by fumarase. And this is a step which takes place by the addition of water, and this is also a reversible reaction. And the L-malate which is formed here, it's going to be the kind of stereo-specific compound, because the enzyme which is going to be followed by the next step, it's very stereo-specific. Okay, so the enzyme is malate dehydrogenase, which again act upon the L-malate and converts it to oxaloacetate. So thus the oxaloacetate which was utilized in the first reaction, it's being replenished in the last step of this Tca cycle. And so we can see that here as the end of this cycle, we can conclude that it has formed 3 NADH molecule and 1 FADH2 molecule is also being produced, and along with this 1 ATP or GTP molecule is also made in this cycle. So on the whole, we can say that this acetyl carbon atom, whatever has been utilized in the earlier step, that has been liberated in the form of carbon dioxide. So the acetyl CoA which was utilized, which entered the pathway in the previous step or previous turn of the cycle, that is released in this turn as carbon dioxide. So the acetyl CoA which has entered the pathway in the current cycle, that will be released in the form of carbon dioxide in the latter cycle. Okay, so that's how this entire pathway proceeds. Okay, and it is also going to be an anaplerotic reaction, because whenever the Tca cycle is going to be deficient of oxaloacetate, we see that this pyruvate is carboxylated to form oxaloacetate via gluconeogenesis as well. Okay, so we see the enzyme pyruvate carboxylase. It has the ability, or it converts pyruvate to oxaloacetate in an intermediate step, and this oxaloacetate can be pumped into the Tca cycle as well. So that's why this is called as an anaplerotic reactions. Whenever there is a deficiency of the intermediates required for the Tca cycle, it can be utilized from some other pathway. And this Tca cycle, it does not generate a large amount of energy as such, but when this particular pathway is coupled with oxidative phosphorylation, it generates more amount of ATP. So as a result of this oxidative phosphorylation, we can see that 2 molecules of pyruvate which is formed through glycolysis, it gets converted to 6 molecules of carbon dioxide, which ultimately yields 32 ATP approximately. Okay, so 32 number of ATP which is produced in this Tca cycle, it results in the production of 976 kilojoule per mole of energy. So that is how much of energy that is produced via this Tca cycle. So next we move on to the regulation of this Tca cycle. The first main regulator is the PDH enzyme, that is pyruvate dehydrogenase enzyme, which catalyzes the conversion of pyruvate to acetyl CoA. And the second one is the step which is catalyzed by the enzyme citrate synthase, and followed by the isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. So here this first enzyme PDH, it is inhibited by ATP, acetyl CoA and NADH, and also allosteric inhibition by long chain fatty acids is also seen in this particular pathway. And whenever there is a scarcity for this acetyl CoA in moiety in this particular pathway, this enzyme PDH, it gets activated, along with AMP, CoA and NAD+ also acts as activator for this particular enzyme. And when there is sufficient amount of acetyl CoA is present in this in the cell, which is derived from fatty acid metabolism, Tca cycle will be turned off, and also this PDH enzyme will be inhibited by reversible phosphorylation of any of the subunits of this enzyme, and this happens when sufficient amount of ATP is present. So this can be reversed, or the pathway can be turned on by the enzyme phosphatase. This enzyme dephosphorylates the subunit of this particular enzyme, and that happens whenever there is a decline in the amount of ATP in the cell. And the next one is citrate synthase. Here the main the one which is going to limit this particular enzyme is the availability of the substrate synthase. And a high NADH plus or the increase in the reduced form of NAD, it inhibits most of the dehydrogenases, and product inhibition is also seen for these enzymes. So whenever sufficient amount of product like citrate or alpha ketoglutarate is present in the particular cell, the pathway will be turned off. And under normal conditions, we can see that the rates of the glycolysis and TCA has been integrated, because TCA has to obtain the pyruvate only from glycolysis. And we also have seen that the enzyme phosphofructokinase, it's been inhibited by citrate. Okay, citrate is a intermediate present in TCA cycle, and phosphofructokinase is the enzyme which is going to phosphorylate the fructose which is formed in the first step of glycolysis, or sorry, second step of glycolysis. Okay, so we can see that there is a very close integration is seen between these two pathways. So with this we come to an end of this TCA cycle. So we will catch up with the next topic in the next video. Thank you.